This is Jim Folsom. Thank you, Jim, for your patience. We had a little snafu with the registration and the getting in there. So without further ado, Jim has been the curator there at the Huntington Gardens in San Diego, so we're excited to have him. He started his adventure with plants back in graduate school, and with this orchid actually in the Apalachicola Forest. So Jim, take it away.
Okay, well, can you hear me?
Yes.
Can you hear me?
Yes, we can hear you.
So I'm okay. Then I'm gonna speed it up a bit because people have been waiting. So every plant has so many stories that could be told about it, but Chapman's orchid is a special plant in that regard. And I'm going to walk you through a series of stories and some ecology and some evolutionary biology, just lightly. And let's start with the plant itself. Here's an old photo that I got a scan program and scanned it from a slide. So this was taken back in the 70s. And that's just a lovely specimen of this orchid that's mostly native to the panhandle of Florida. The core distribution is right here in Apalachicola National Forest. We'll look at some distribution maps in a minute. Chapman's orchid was first apparently collected by a fellow who lived right here in Apalachicola, Dr. Alvin Wentworth Chapman. And you see that on the screen that his name is spelled Alvin by most people, but if you go to Wikipedia, it's spelled with a V-A-N. So you'll find him under either or both. And in every case, you'll find a very short synopsis of his life. He wrote his own biography, which is these 2 or 3 paragraphs right here. So that's his whole life story as he told it. What you're seeing on the right is Charles Moore's obituary for him. Now, am I still there? I just got something closed. That was the old one, maybe. Am I still with you, Anita?
Yes, yes, you're here.
Okay.
We got you.
Because I just got a message saying I was thrown out. Okay. So you're looking at a picture. So who was Alvin Wentworth Chapman and how did he come to know this orchid? Well, he wrote the first flora of the southern states, and he moved to Apalachicola in 1847. So he was there throughout the entire time of the Civil War until his death in 1899. He published his flora, and you can see here's some statements about him. He was a very gentle, mild man. He was a doctor that got his training in Kentucky, though he himself grew up in Massachusetts and is one of the early graduates of Amherst College, probably one of their earliest graduates with any notoriety. So here's the front page of the flora, 1860. There were 3 editions, 1884 and 1897. So if you think about that for a second, his flora was published a year before the Civil War, or the war between the states, as we called it in Eufaula. So he published his flora, and it was huge. It was 600 pages. So it was an astonishing work that that took most of his sort of academic and field effort. He dedicated it to this fellow who got a lot of specimens for him. And if you look here, just in small print, you'll see that he treated Platanthera as the genus Platanthera. Historically, this group of orchids has been treated as Orchis by Linnaeus, as Habenaria, as Platanthera, and then once again as Habenaria, and then by Rafinesque as Blephora glottis, and finally back to Platanthera. So as far as I'm concerned, you could just call it whichever genus you wish, with Blephora glottis, Platanthera, Habenaria, but pretty much Habenaria is the recognized genus anymore. So this is Chapman. He knew this plant was different. He collected the specimen that was selected by John Kunkel Small. In 1903, Small published his flora of the southeastern U.S., and in that he published the Platanthera chapmanii based on a specimen that Chapman had sent to Columbia College, because Chapman was sending specimens to two places, one of which was Harvard because he worked with Asa Gray, and one was Columbia College. Columbia's herbarium was dismantled and given to the upstart Botanical Garden in 1890, the New York Botanical Garden. And with the herbarium went this young fellow, John Kunkel Small, who was at New York Botanical Garden practically until his death in 1938. So Small then writes his flora of the southeastern US, published just 4 years after the death of Chapman. Now, Small was an imposing character. Here's his flora, 1903. And you got to remember something incredible. Back in those days, even though this was a huge flora, over 1,000 pages, Small had to finance it himself because New York Botanic Garden was new, and the gardens at that time did not pay for your publications. So both Chapman and Small had to self-publish their floras. So here's the title page for Small's for 1903, which he revised in 1913. And here you can see he's accepting Rafinesque's genus, Blephara glottis. See that little accent mark above the O? What's really fun about that is Small was so careful. He wanted you to know where to put the accent. So he wanted you to say Blephara glottis instead of Blephara glottis, and he wanted you to clearly say Chapmanii. So on all of his florist, all of his names, he gives you a hint as to how he expects you to pronounce this word. Look at his key. He keys out these orchids together: Cristata, Blephara glottis, Conspicua, Ciliaris, and Chapmanii. And he keys them out based on the lip not being three-lobed, which is really curious because the thing that holds them together even more thoroughly, except for one form of Blephara glottis, is that they are highly fringed. The lips are fringed. So he keys all these together, and there's Chapmanii, his publication of it. So here's number 5, Blephara glottis Chapmanii, and he describes it. And the key difference that he uses to separate it is pretty interesting. It's the fact that the petals, we'll look at this in a second, the petals are wedge-shaped, cuneate, rather than linear. And that's an interesting, I remember noting that, but it's interesting that that's how he does it. So Small passes away in 1938, a big guy with a big reputation, and this is the closing statement on his obituary. The total of his published books and papers is unusually large, and he left many manuscripts, complete and incomplete. But his greatest weakness was the tendency to exaggeration for the sake of emphasis. Yet, he was so careful in his work that few of his statements can be questioned, and future studies will serve only to confirm them. I'm here to actually confirm one of his many statements, and that is that after Small's publication, along came the period of biostudies in taxonomy. And so biosystematics involved a lot of concern about plants being hybrids. And a young, very wealthy orchidist at Harvard named Oakes Ames had named a hybrid Canbii, which was the hybrid between the white fringed orchid, that would be Blephara glottis or Platanthera Blephara glottis, and Ciliaris. So he had named that and started looking about and noted that Platanthera Chapmanii, which Small he called Blephara glottis Chapmanii, was intermediate in character between the large Platanthera cristata, the large Platanthera Ciliaris, and we'll see photos of these, and the small Platanthera cristata. So he decided to designate it as a hybrid, and it stayed that way until I went to Vanderbilt in 1975, and Ben Channel, you can see his name, he's my major professor. Ben Channel had collected this hybrid in a mixed population, and he was curious about the nature of this hybrid. So I started looking into it and took it on as my thesis. And so here you see my camping apparatus, and this is about 1975, I had a little 4-cylinder Chevrolet called a Monza, and it was a hatchback. It was great for camping out, and I drove the forests of Apalachicola for 3 summers in a row, sleeping at Wright Lake, running into all the denizens of the forest and working with these orchids. In fact, I even did chromosome studies, and there's a field Nikon microscope and a Coleman lantern and a CO2 tank, because you had to capture, you had to work with the chromosomes between 7 p.m. and 10 p.m. to see meiosis. They had this kind of early nighttime crepuscular meiosis. So I was trying to see if these things had done that wonderful trick of becoming tetraploids, which I discovered they had not. They all have the same chromosome number. So that gets us to the plant itself. Here are some plants of Chapmanii. I moved them all over the place. I dug them and hybridized them with all of their associate species to make sure that I had no barriers to hybridization. And I, with the permission of the Forest Service, kept a population along Road 100. What's curious is that population is totally gone today. And I think that's partially because I put a sign up that said biological research or experimental site, and came back a few years later and people were using it to plant marijuana. So it's a huge marijuana farm, which I guess they figured it was protected by that sign. And I think it must have been sprayed out. Everything got sprayed, because there's not a single, out of several hundred orchids at that site, there's not a single one that crops up today. So I'm guessing somebody went in with herbicide and wiped out everything there. But anyway, so these are some of my experimental plants. And here's a population along Road 65 that you will see now every summer. And that population, when I first encountered it in 1975, was about 5 plants. And today, that population stretches from Toloja all the way to Sumatra, concentrated around the intersection with Road 100, Forest Road 100, but scattered in small little outposts all along the highway. I personally think that what's going on is the flowering of these plants, they get mature seed just before Thanksgiving. Somewhere in October, they have mature seed. That's when the county crews are allowed to do their more distant cutting. And I think, curiously, what's happening is the county crews are dispersing, using their equipment, they're dispersing the seed, the mature seed of this orchid all up and down the roadway, which I'm not complaining about. I understand from Lauren Anderson that something similar seems to be happening to, was it what's the genus, the lily genus? Not Harperella, but it'll come. Anyway, so there's a curious thing happening along the roadway. Here's that population of Platanthera chapmanii. Here are some flowers up close. That is the orchid column. It doesn't look like the face of some kind of an insect. These auricles are sort of interpreted as being the where 3 stamens, 2 other stamens might be attached if they existed. But all of these orchids, in fact, almost all orchids except the Cypripediums and the early apostonoids, have only 1 anther. So this structure is the 1 anther broken into 2 sacs. The anther structure of an orchid is called a pollinarium, but each of these is called a pollinium. We'll look at that more closely in a second. So this is the 2 halves of 1 anther in this orchid, looking right into the face of the orchid. Right under there is the entry to the nectar, the spur. So you can see there's the nectary there. And the entrance is in this petal, which is called the lip. These are the other 2 petals. As a monocot, this thing has 3 petals and 3 sepals. That's a sepal, and folded down below the flower are the 2 lateral sepals. That's a petal, that's a petal. And here's the 3rd petal, called a labellum. It sits exactly opposite this structure, which is the anther, combined with, in this cavity, the stigma. So that, of course, is the definition of an orchid, is a single anther combined with the stigma and a structure called the column. So let's look a little bit more. Now, here's a picture of Chapmanii from the side. There are the two halves of the anther. And you see, if you can see it, there's a little disc right there. That's called the fascidium. It's sticky. Here's some more pictures. There's that disc. So each of the pollinia has this thing called a stipe and this sticky disc. Doesn't it look like a bird in this picture? It's really, here it is again. Notice how the column bends down right over that orifice. So here's the orifice to the nectary. There's the nectary right there. Okay, so let's start over. This is the attachment. This is the ovary, which is inferior. Here's the dorsal sepal, a lateral sepal. Here's the lip. At the base of the lip is the opening to the nectary, which is almost at the same length as the ovary. This is the anther, and that's one of the fascidia. So the pollinator would land on these fimbriate edges to the lip and would probe the nectary for nectar, pushing its proboscis right past these two sticky discs. So here's a population again, and I spent hours, days at this population this summer, hoping to film live pollination. And what was curious was I only saw one pollinator in several days, whereas in 1975, I could go to the population every day and easily find pollinators, which you'll see in a minute are big papilias. So let's just keep going. Now, this plant, Platanthera chapmanii, has a curious distribution. Here it is in Florida. As Lauren tells me, it misses this wet basin from the Swanee River. But notice it's got a disjunct part of the population way over in Texas. That's part of the many stories that relate to Platanthera chapmanii. Here we have the chapmanii again. Once again, looking at the flowers. So we look straight into the flower. There's the two discs. This is the anther. I've dislodged this. I've poked in there, and you can see one of the pollinia dislodged with its sticky disc. Here it is right here. It looks like a double exposure. It's not. That's a dislodged pollinium. Here it is. So you can take, this is a pine needle. Take a pine needle or your fingernail or a probe of some sort, and you can insert it in the flower, and the pollinium will stick to that object. And when you pull it out, this is what you get. So you get the basidium, the stipe, and a mass of mealy kind of pollen. It comes out sort of erect, and over time it bends down. So that's the same pollinium after a minute or two. Here it is in size. So there it is against my finger. It's remarkably intricate as far as structure goes. And this is one of the great things about orchids, especially the more advanced orchids. They have this incredible, highly structured, intricate, these pollinaria that are part of why the pollination systems for orchids are so elaborate, or can be so elaborate. So that's Platanthera chapmanii. And here it is, a picture, once again, not so good because I used one of those. I used my phone and captured a picture from an old slide, of which I have hundreds. This is the visitor, a papilio, and it's probing a flower. Now look at the next picture. That is the pollinium attached to the proboscis of the butterfly. So over and over, I was able to document that this is how Platanthera chapmanii is pollinated. It has a short spur that, because the fascidia, the column is bent the way it is, when the butterfly inserts its proboscis, the pollinia attach to the proboscis partway down, like anywhere from 7 to 10 millimeters from the tip of the proboscis. And I didn't just see this once, I saw this over and over and over again. So I was pretty convinced in several populations that what I was looking at was a plant with a pollination system in which the shape of the column was absolutely tied to how this plant was pollinated. So let's look now at the rest of the story. This is once again, the slide itself is beautiful. I'm sorry, the translation to digital is so bad. So here we have three plants suddenly. We have Platanthera chapmanii. We have the more widely known Platanthera ciliaris, the yellow fringed orchid, and the even more widely spread, but not as well known, Platanthera cristata. So Platanthera cristata, Platanthera chapmanii, and Platanthera ciliaris. So ever since Oakes Ames came along, this plant was considered the spontaneous hybrid between these two. And indeed, if you go to Tennessee or you go to a place where you'll find a mixed population of ciliaris and cristata, you might well find one or two of these. I documented them all over the United States, the eastern United States. What is different is in Apalachicola and across the panhandle of Florida, this form exists in populations. And that was the big aha moment for me when I'm doing all of this library work, and I've borrowed specimens from thousands of specimens from around the US and I've inspected them all. And then one day I'm sitting here near Apalachicola in the forest and I'm thinking, wait, wasn't this plant named for Chapman and he lived here in Apalachicola? And it was like I felt stupid for having put off that realization. But suddenly I thought, oh my gosh, the plant Chapman knew is not exactly the same thing as another plant, which is the spontaneous hybrid that you might find anywhere in the country. So let's keep talking about this. So now I'm interested in documenting what's going on here. Here's my drawing of Ciliaris, chapmanii, and cristata. And here's a more kind of almost lurid drawing looking straight into cristata, chapmanii, and ciliaris. Notice that the Visidia in ciliaris, and we'll see live photos of this in a minute, are really flayed out. And ciliaris has this huge, that's the stigma. So there's one pollinium in there, another pollinium in here. So that's one anther, right? And then the stigmatic cavity. Here's looking straight at chapmanii. The two Visidia kind of knuckled down and folded together. And there's the stigmatic cavity, which is sort of these pouched lobes looking a lot like the stigma of the smaller flower of cristata, which has the Visidia looking straight at you. And here's profile. So this is cristata, chapmanii, and ciliaris. Let's look at so here's ciliaris. Much bigger, much floppier, casual, more showy. There's its distribution in Florida. Notice that it is not, I've never seen a collection from right where we are. It's like bizarre, but there are a lot of plants that show this curious distribution. And here's a closer up of the flower. So there's the two pollinia. There's the sticky disc right there. So this is the flower of Ciliaris, almost twice as big as the flower of chapmanii. And here's what happens when you pry out that pollinium. I've taken, it's a bigger flower, and I was younger then, and with a sharp probe, I was able to push out that pollinium. That's the sticky disc of this one, and this one you don't see anymore. But this is the pollinium. Here it is stuck to a penny. So that's the pollinium of Ciliaris. And after a while, actually a long while, I let this stay on the penny for a month or so, it eventually collapses down. So that's the pollen. This is the stipe, and there's the sticky disc. It's just an amazing structure. And here is the same butterfly, or at least another Papilio, visiting Ciliaris in Russell County, Alabama, just south of Phoenix City. And that's Ciliaris. Look at the butterfly's eyes. Here we go again. So if you visit a population, Platanthus ciliaris, in full bloom, it's pretty common to run into these big butterflies. And what you'll find is that the nectary is sufficiently long such that the butterfly has to dip all the way into the flower to get to the bottom of the nectary. The two anther sacs are splayed outward, and the visidia stick to the butterfly's compound eyes. I have found butterflies with as many as 15 or 20 pollinia covering their eyes. I didn't even know how they could, with those compound eyes, how they could even get around. So the pollination system for Platanthus ciliaris is different. The butterfly does the same thing. It lands on these fringes, it probes the nectary, but it has to go deep enough to where the pollinia, which now are positioned facing outward, the sticky disc, get plastered all over the eyes of the butterfly. So that's a different pollination system. There are problems with this. The problem biologically is that it's unlikely you will ever get pollen from a ciliaris into the stigma of a Chapmanii, but the reverse is more easily assumed, and I actually tested this, and I think it's true. If you had a mixed population of ciliaris and Chapmanii, you would see a regular transmittal of pollen from the proboscis of the butterfly into the stigma of ciliaris. So I think it's not a perfectly, completely segregating isolation system. Okay. Now, it gets more complex because this is ciliaris, see, with the anther sacs kind of straightforward displaying out. There is a white version of ciliaris. So there are several, and they are exactly the same except shorter fringe and white and fragrant at night. Ciliaris is never fragrant. This is fragrant at night. So I visited populations of this, and this is pollinated, as you would suspect, by sphingid moths. And there are various forms. There's one with a very long nectary. There's a form that has no fringes whatsoever. There's this form, conspicua, because it's, so there are like 3 forms that have all been named. In my mind, they're pretty much all the same thing. They may be different varieties. You could call them different species. But it's the white flowering, the night pollinated or crepuscular pollinated version of ciliaris. And in many older treatments, this was just a subspecies or a variety of ciliaris. But it's clearly pollinated differentially by a moth. I captured the moths. I studied that enough to understand, to be comfortable with the fact that these 2 plants are separated by pollination system. And then we get to the other potential parent of these plants, the small one. This is cristata. And even though I've seen more cristatas than any other plant over my history, and remember, this was all done back in the 70s. I'm just reawakening, like Rip Van Winkle, to the interest in these plants. So this is cristata. And look how the 2 anther sacs look straight forward. There's the entrance to the nectary and a short nectary. I never successfully confirmed the whole pollination system for this, even having spent scores of hours in populations watching. My assumption is this is probably mainly bee pollinated, but I captured every critter in the world, different sized butterflies, the big ones, smaller ones, even bees visiting. I only one time found anybody with the plainia on some body part. So There's still work that could be done here with this funny little plant cristata. And here's its distribution. So it does occur where we are. If we look at the map I generated back in my thesis in 1979, this is the whole complex. And Ciliaris is the little round circles. Cristata is the little dark filled in circles. And then if it's a triangle inside a circle, it's what I call Chapmanii. Populations here, there were some here, and there are some here, and then way over here. What the heck is that all about? But I segregated in this any individuals I found that were in mixed populations and not part of this. And so what's the difference between those? Here we go back to that picture again. There's that bent hammer toed column of Chapmanii. And here's the difference. If you inspect flowers from Florida and Texas, you will find this column shape consistently. It varies a bit. They can be a little bit further apart, but you will find the two Visidia not only bent down, but approaching one another. However, if you find a plant out of this southern tier of the coastal plain, up in the mountains of Tennessee in mixed populations of Ciliaris and Cristata, the column would be what you expect the column to be. It will project forward, and the two halves of the anther will not converge. They'll flay away from each other. So these plants are absolutely interpretable as hybrids between the bigger Ciliaris and the smaller Cristata. And they have been conflated with these plants along the coastal plain, on the southern tier of the coastal plain here in Panhandle of Florida principally, that may have begun as some hybrid historically, anciently, but through selection over time, these have adopted their own pollination system using the same butterfly. It's all very curious. And what about Texas? One could even speculate that this has happened twice, that the plants here are not exactly the progenitors to the plants in Texas. It's possible that in the Big Bend of Texas, where the flora resembles the flora here, on the other side of the Atchafalaya swamp, it's possible that you could have gotten some of these plants transported, the seed transported by birds. So it's not impossible that the Texas plants are derived from the population here. It's also not impossible to imagine that the same thing happened there, that there was a new hybrid and it was also selected. So there's a woman right now doing her master's work, I think, in Texas, studying the DNA, the heritage of this plant. So it'll be curious to see whether this plant is derived from some population of Chapmanii here in the east, or whether it really needs its own name as a separately evolved species. And that would be a unique circumstance. So just a few years ago, way after my work, Paul Martin Brown from University of Florida went in and looked at the whole complex, and he named everybody else. So here's Platanthera chapmanii, here's Platanthera cristata, Ciliaris, and the white Blephoroglottis. Now, what I did was I said, look, Chapmanii is a species, but the hybrid exists, and I'm going to name that for Ben Channel since it was his idea. So that's Platanthera X, or it's not really a time sign, it's not really a Chanellii. And so Martin has named Osceola, Apalachicola. Canby was already named by Ames. Bicolor had been named. He named the hybrid between this form of Blephoroglottis conspicua, Lurii, after Carlisle Lure, who did the orchids of Florida, and he named this one between conspicuous. So now there is this remarkable complex that looks like a great big tabernacle or a tent, and everybody's invited to the party. So back to Chapmanii, you will encounter it in populations that thankfully some have grown, others have disappeared. Its natural habitat seems to be in those grassy understories to tall pine forest, but it doesn't mind roadsides at all. It's, in fact, neither does Ciliaris. So here it is looking straight at it. I got some great hints from people here. David Roddenberry pointed me to new populations I had never seen. There's a population in the forest south of south of Tallahassee there along the coast, and it's the furthest south I've ever seen it, and there are further populations over near Carabel. What's really curious is it doesn't exist west of pretty much the Apalachicola National Forest. It doesn't exist all the way from there to Texas. And I have driven those roads a lot and looked, and you just will not find it. At least I never found it. So I think that's yeah. And just one last photo of that curious shape. So if this were the hybrid itself, the de novo hybrid that occurs spontaneously in populations, that would not be curved. It would look straight out. And so when you see that, if you're out in the field and you want to identify Chapmanii, there are a lot of clues. One is the spur, the neck tree, is about the length of the ovary. The column, the two halves of the anther converge and bend down. And then the last one is that thing from the key that our friend John Kunkel Small wrote. If you look at a dissected specimen, you'll see that the lateral petals, this is a lateral petal, that's a sepal, that's a lateral petal. In Ciliaris, they're sort of linear, strap-shaped, but in both Chapmanii and Cristata, they're very wedge-shaped. So you can use that too. Anyway, I think that's pretty much all I have to pass on to you, and be glad to entertain some questions. Anita, how do I get out of this?
Just, you can, I sent your webcam.
There's an orange arrow. Is that what I do?
Just,
let's see. Stop showing screen.
Yeah, and you can you should be able to enable your camera.
That's
great. There you are. Hi. Thank
but I
you
can't
so
see you.
much.
So.
Okay. Yeah, I have my camera off, but thank you so much, Jim. That was wonderful. They are hauntingly beautiful. Does anybody have a question? You can come off mute or raise your hand or put the question in the question panel. There's a lot to take in.
And I've sped it up because people have been waiting so long.
I know, we appreciate it. I'm sorry for all the technical difficulties we had at the beginning. We just weren't able to make that meeting work. So I appreciate everybody pivoting and getting on the new call. I'm not seeing any questions or hands up. Oh, wait, here we go.
I got it.
Let me pull this up here. Has anyone tried to do any molecular phylogenetics with this group?
Yeah,
that's
there have been a couple of attempts, but the woman in Texas is working on that right now. What I don't know is if she plans to compare Chapmanii in California. I've been in California. Chapmanii and the Panhandle with the Big Bend populations, okay, the big thicket populations. But I wrote her a long note and suggested things that would be really fun to know. So
Great. Any other questions? Scrolling. Let's give it a minute here.
Yeah.
Okay. Not seeing any other questions. You have any other points? Anybody else? Okay, well, thank you so much. Sorry, like I said, I'm so sorry we had the earlier problems. I really appreciate everybody, including our speaker, being so flexible and able to change out. Our next Sci Cafe will be April 28th, so we hope you will join us. We're hoping to get Renee Colini to talk about the new NOAA models, sea level rise models. So Jim, we thank you so much. And Jim is also going to serve on the, tell them your role with the Florida Wildflower Foundation.
yeah, and I would appreciate any input or help. I volunteered because I'm fresh meat, I'm new, so I volunteered to be the county rep for both Franklin County and of course for Liberty County, which is not really part of the agreement. But I'm so fascinated by Liberty County and I have done so much work there. And what I'm supposed to be doing is communicating with the people who do all of the cutting of the roadsides, the mowing. And there are agreements that exist between different parties and the different counties that protect certain stretches of roadside from being widely mowed during the certain times of the year, such that you delay the furthest out mowing in certain stretches to allow reseeding and to allow plants to flower. And so the question is, how well do people understand how that works, and how supportive are agencies? So far, I see some pretty good compliance and support. But on the other hand, there's a huge amount of educating the people who do all the work as to how incredibly fascinating this flora is. I mean, I don't think it's impossible to overstate the unique significance of the flora here in our region on a national basis, on a world basis. And I wish people could really fall in love with how important the natural history of this flora is, and how worth preserving that is, worth protecting. I live on the island, and it's not just the road shoulders that concern me. I watch people, everybody wants to clear the property, any lot on St. George Island, and make it look like some tropical paradise, which is not paradise at all. It's a flat, cleared lot with full sun and a house sitting in the middle of it. Whereas in reality, paradise has been here for a long time. The incredible flora that we have that makes as good of a yard as any number of hibiscus and oleanders that you can drag in from a nursery. So I'm hoping that we can convince people to really fall in love with the native plants and give them a chance to survive here.
We need to do another Sci Cafe aimed at those people so we did keep our.
I mean, we just need the realtors to up the value of your property. I wish the state would pass some law that would give you a somewhat of a discount on your property tax if you preserve native plants around your home. You know, something like that would be cool as heck.
That would be a
great,
great project for the Florida Wildflower Association.
Well, they're always cutting taxes, right? So if they want to cut taxes, let's cut taxes for people who do great things. Yeah. Well, anyway, thank you very much. It's nice to have talked to the group, and I appreciate any input.
Yeah, just quick, we had another question from Susan Farmer. She was asking, would you give out your contact information?
Sure.
Okay.
You can, Juanita's got it. I'm jfolsom@huntington.org, and that's Folsom as in the prison. It's FOLSOM. But I also put out I'm the most nerdy, boring person on TikTok. So you can find me on TikTok as Botany in Context, one word, Botany in Context. And since May of last year, I've produced is not the right word. I have haphazardly recorded over 1,000 videos, and a lot of them are right here on the island and up in the forest. So I appreciate people seeing those and giving me input on those. And that's a good way to touch base with me, because a lot of people in the comments say, oh, you wrongly identify that. Oh, by the way, we don't have Sarracenia purpurea now. We call it everything here. We call Sarracenia rosea. And I'm thinking, like, it sure looks like purpurea to me. But anyway, so I get a lot of great comments and I learn a lot. Lady Bird, that's one of your state forest people. She has given me good name, good identification on several plants, and her TikTok account is really cool. She posts some incredible things about the native plants here. So
Great.
So that's a way to get in touch with me.
Well, thank you so much. You are a wealth of knowledge, and we look forward to helping you spread the word about native plants for sure and how valuable they are. So we'll have you back soon. Well, thank you everybody for joining in, and we will see you at our next talk. And thank you so much, Jim.
Goodbye.